Hello everybody, in this video I'm going to talk about the physiology of heart muscle contraction. First of all, we should know that the myocardium, like smooth and skeletal muscle, responds to stimulation by depolarization of the membrane, which is followed by shortening of the contractile proteins and ends with relaxation and return to the resting state. Here's a deep look on myocytes. And what I mean by myocytes or the myocardial cells, okay? However, unlike skeletal muscle, which shows graded contractions depending on the number of muscle cells that are stimulated, the cardiac muscle cells are interconnected in groups that respond to stimuli as a unit, contracting together whenever a single cell is stimulated. Okay, when we want to talk about action potential, we should know this idea. Cardiac muscle cells are electrically excitable. However, unlike the cells of other muscles and nerves, the cells of cardiac muscle show a spontaneous intrinsic rhythm generated by specialized pacemaker cells located in the sinoatrial and atrioventricular nodes. If we take a look on this figure, we have many pacemaker cells. We have SA and AV nodes, we have His bundle, we have Purkinje fibers. Each type of these pacemaker cells is located in a site within the heart, as we see here. The cardiac cells also have an unusually long action potential, which can be divided into 5 phases. We have here a figure that illustrates the major ions contributing to depolarization and polarization of cardiac cells. When we take a look on this figure that represents action potential of a Purkinje fiber, first of all, we can observe two axes. One of them is vertical and represents the potential by millivolt, and the other is horizontal and represents the time by seconds. So um, when a time which is between 0 and 1 second, in each second we will have this curve which is in the red line. So what's happening is happening within 1 second. This is the first thing that we should concentrate on. The second thing, within this 1 second, we have changes in the potential in millivolts from in range, which is minus 50 to plus 50 millivolts. We also have 5 major phases: phase 0, phase 1, phase 2, phase 3, and phase 4, as we see here. So as we see here, these 5 phases occur within a second and represent the changes in the potential measured by millivolts. Now we will begin with the first phase, which is phase 0, and it actually represents the fast upstroke. Here, first of all, we should know that we have a heart muscle cell. This cell, there is a change in the concentration of positive ions or cations between the inside and the outside of this cell. And these changes in the charge inside and outside the cell will lead to the potential, and as a result we will have a contraction of heart muscle cell. So if we take a look here on this figure, which represents the cell membrane, we will have as we see, we have outside cell and inside cell. from the outside into the inside, there is a flow of sodium positive ions or cations. So here, this flow is due to the that we have sodium channels, and these sodium channels open. The result is this flow. Here, after that, this upstroke ends as sodium channels are rapidly inactivated. So sodium current is blocked by antiarrhythmic agents such as quinidine. If it's a note here. The third point is a note. So what we should know, that the beginning of our story is in within phase 0, where sodium ions enter the cell as a result of um, the activating of sodium channels. Then we have this phase, which is phase 1. When we take a look another time on the red curve, uh, within uh, within the flow of time, okay, here, as we saw in the previous slide, when sodium cations enter the cell from outside into the inside, here, before this process, we have the inside uh, segment or part of cell membrane is highly negatively charged. So when these cations or positively charged ions enter from the outside into the inside, so here, as we see, we were in a point which is bigger than minus 50 millivolt, and then within the phase 0, we will have this increase from negatively charged to positively charged uh, inside segment of this cell, as a result of the entrance of these positive ions into the cell. Now here in the phase 1, as we see, the sodium channels are inactivated, and the inside portion of cell membrane is highly positively charged. So we should have a balance. A balance is represented by the exit of other positive ions than sodium cations or positive ions. So here, these alternatives are potassium ions, which are also positively charged ions or cations. So they will go out from the inside, go out from the cell. So the flow here is from the inside into the outside. So the initial rapid phase of repolarization here is due to inactivation of sodium channels and potassium channels that rapidly open and close, causing a transient outward current. So this is the main case here. So after that, we will have this phase, which is the phase 2. It represents something which is called the plateau. And here, as we see in the red curve, within the red curve, here the voltage or the negatively charged ions will increase more and more. So this will be as a result of the exit of potassium ions and the entrance of calcium ions instead, as a result of the opening of something which is called the voltage sensitive calcium channels. So after the repolarization, we will have this depolarization. So these voltage sensitive calcium channels open, resulting in a slow inward depolarizing current that balances the slow outward or polarizing leak of potassium ions. So it's about balancing. We started in the entrance of sodium, and then this stopped, and we will have the exit of potassium, and now we have the entrance of calcium instead. All of these things are just to do the repolarization and depolarization. Then we will have this phase, which is the phase 3. It represents the repolarization. Here, the calcium channels close, as we see in the figure, and potassium channels open, resulting in an outward current that leads to membrane repolarization. The net result of the action to this point is a net gain of sodium and loss of potassium. This imbalance is corrected by sodium-potassium ATPase or adenosine triphosphatase. The last phase here is phase 4, which represents forward current. Here, increasing depolarization results from a gradual increase in sodium permeability, and the spontaneous depolarization automatically brings the cell to the threshold of the next action potential. So this is the process of these four or these 5 phases: phase 0, phase 1, 2, 3, and 4, that represents the depolarization and repolarization and the acting of the channels and the entrance and the exit of these ions, which are potassium, sodium, and calcium ions within the two faces of the cell membrane of course heart muscle cells. These ions pass through channels in the sarcolemmal membrane and thus create a current. The channels open and close at different times during the action potential. Some respond primarily to changes in ion concentration, whereas others are sensitive to adenosine triphosphate or to membrane voltage. We talked about the muscle contraction process, and now we will concentrate on the cardiac contraction. The contractile machinery of the myocardial cell is essentially the same as that in striated muscle. This is why I told you in the previous slides that we can consider these cells as cardiac cells. Okay, the force of contraction of the cardiac muscle is directly related to the concentration of free or unbound cytosolic calcium. Therefore, agents that increase these calcium levels or that increase the sensitivity of the contractile machinery to calcium result in an increased force of contraction. And here we can say that this represents a term which is inotropic effect. And we should note something, that the inotropic agents increase the contractility of the heart by directly or indirectly altering the mechanisms that control the concentration of intracellular calcium. So the free intracellular calcium here is important. Actually, what are the sources of this free intracellular calcium? Calcium comes from several sources, by the way. The first is from outside the cell, where opening of voltage-sensitive calcium channels causes an immediate rise in free cytosolic calcium. Calcium may also enter by exchange with sodium. Calcium is also released from the sarcoplasmic reticulum and mitochondria, which further increases the cytosolic level of calcium. Another important point here, which is the removal of free cytosolic calcium. Actually, if free cytosolic calcium levels were to remain high, the cardiac muscle would be in a constant state of contraction rather than showing a periodic contraction. Mechanisms of removal include two alternatives: sodium-calcium exchange, and on the other hand, uptake of calcium by the sarcoplasmic reticulum and mitochondria. Okay, what about sodium-calcium exchange? Calcium is removed by a sodium-calcium exchange reaction that reversibly exchanges calcium ions for sodium ions across the cell membrane. This interaction between the movement of calcium and sodium ions is significant because changes in intracellular sodium can affect cellular levels of calcium. When we want to talk about uptake of calcium by the sarcoplasmic reticulum and mitochondria, we should know that calcium is also captured by the sarcoplasmic reticulum and the mitochondria, and more than 99% of the intercellular calcium is located in these organelles. And even a modest shift between these stalls and free calcium can lead to large changes in the concentration of free cytosolic calcium. Okay, here we can take a look on what happens actually within the two sides of the membrane of cardiac muscle cell. Here we have voltage sensitive slow calcium channel and sodium calcium exchanging, and we have also sodium potassium ATPase pump. Here, first of all, calcium entry from outside the cell triggers the release of a much larger quantity of calcium ions from the sarcoplasmic reticulum. Then this will lead to increased calcium concentration, which initiates the contractile process. Then calcium is removed by reuptake into the sarcoplasmic reticulum and by extrusion from cell by calcium sodium exchange. The final step here, which is the fourth step, is that sodium balance is restored by this sodium potassium ATPase. So uh, um, we can say that it is very it likes, or it's very close to what happens actually within the muscles, the mechanism that I talked about, and the phases from 0 to phase 4. So it's actually very close to that mechanism, and this is actually what happens within the two sites, and this exchange between ions and the effect of calcium and how the cell removes it. Okay, so everything is summarized in this figure. Thank you all for watching. Please like and subscribe for more videos.